Good morning. This is Jesse Goodman. I'll be your host today here. I'm currently professor of medicine and infectious disease at Georgetown and formerly was FDA's chief scientist. Welcome to today's webinar. Our purpose is to bring further attention and focus to the ongoing H5 outbreak that's now affecting mammals, birds, and humans, and with a particular focus on human vaccines to help define priorities for what we should be doing now to reduce the risks of a pandemic developing and to prepare for the possibility. We are asking each of our panelists as part of their discussion to hopefully identify 2 to 4 major gaps in actions they see as high priority. And while our focus today is on human vaccines, we realize that similar attention is needed in many other areas, including, perhaps most importantly, preventive and policy work at the agricultural interface to protect workers as well as animals in the farm economy, work to advance global vaccine supply and equity, as well as other medical countermeasures, such as PPE, diagnostics, and antivirals, and work to improve relevant supply chains. In fact, ultimately, vaccines must be part of a much broader, coordinated, and substantively resourced strategy on both H5 and pandemic influenza. From this broader point of view, we also call attention to the recent National Academies meeting on H5 research gaps. And we will have a link to that for you in the chat. And a report will be forthcoming, we understand, even today. However, today we are focusing on vaccines. Whether the current H5 clade spreads among humans or not, we will at some point have another influenza pandemic, and the steps we take now to better prepare us for future flu pandemics as well, and for other complex public health emergencies. We have a tremendous expert group with us today, making up 4 panels. The first, moderated by Georgetown's Rebecca Katz, will set the stage and review the current virology and animal and human epidemiologic situation. The next, moderated by Norman Baylor, former head of FDA's Office of Vaccines Research and Review, will review the science and data gaps on available and investigational vaccines and the state of US and global regulatory pathways. Excuse me. Following that, a panel moderated by Bruce Gellin, former head of the National Vaccine Program, will look at issues beyond the vaccines themselves that are critical in considering their use, including practical issues like communications and building the trust that will be essential if vaccine is to be offered to farm workers and other occupational risk groups or more widely, and whether pre-pandemic or in a pandemic. Finally, a concluding panel of senior leaders, many of whom have led previous outbreak and pandemic responses, will weigh in from their experience, including on broader issues and what they would emphasize to incoming US and other leaders. We're tight on time, so we'll try to keep our panels on track. And we've also included some time for Q&As that the online audience can place and that moderators can address if time allows. Finally, I know that many of us are digesting recent political events and appointments and see uncertainty ahead for public health, including vaccines. I would argue that recent events make it even more important that we define and advocate for the most important public health priorities, and per our discussion of communications, be open to questions and an even-handed dialogue. So again, thanks in advance to our panelists, to colleagues at O'Neill and the Global Health Institute for making the webinar possible and managing its technical aspects. I will now turn things over to our first panel and its moderator. Rebecca, please take it away. Thank you.
Hi, everybody. Thanks, Jesse. So as Jesse mentioned, I'm Rebecca Katz. I'm a professor at Georgetown, and I direct the Center for Global Health Science and Security. I am delighted to moderate our first session today. We have the opportunity to set the tone and do a little bit of scene setting. We are on a bit of a tight timeline, so I want to make sure we have sufficient time. So we're going to really jump straight in. We're going to first hear from Dr. Richard Webby from St. Jude's Children's Research Hospital to discuss the latest on the virology of H5. Well, he'll be followed by Dr. Marcela Uhart from the University of California, Davis, who will talk about the impact on animals, and then over to Dr. Jen Nuzzo from Brown University, who will focus on the human epidemiology. Now, we've asked all of our panelists to provide an overview of these complex topics, and also identify some of the gaps and recommendations that we can build on for the rest of the meeting. And as a reminder, please put any questions you might have in the Q&A box. We'll have about 10 minutes after the panelist presentations to do our best to respond to them. So with that, Richard, over to you.
Thank you so much, Rebecca. Let me just... Okay, hopefully you're seeing that. Good. Yeah, so in the next 5 minutes, what I want to do is give you a 30,000-foot level of where this all started, where we are now, and what this virus is doing. If we want to start from the beginning, the first time that we became aware of at least the phylogenetic tree of this particular virus all the way back was in southern China, way back in 1996, where the precursors of the virus that we're dealing with today were identified in geese in southern China. Again, the first time that we'd seen this particular virus. Although, of course, the virus in 1996 is quite different than the one we're dealing with today. The next sort of major event, that virus, I guess, was detected in 1997 in humans in Hong Kong for the first time. That version of the virus was born in Hong Kong and disappeared with the depopulation of all poultry in across Hong Kong. But the precursors of that virus were still in southern China. Over the years, it sort of tried out different evolutionary paths, and probably sometime in 2023, perhaps a little earlier, a particular combination of gene segments at the time called the Z genotype emerged, and that sort of then signaled the movement of the virus outside of that southern China epicenter to a broader number of countries across Southeast Asia. And of course, this was also associated with human infections across the region, and I think really started the alarm bells ringing for the particular virus. Our next major event was in 2005. Up until this point, it had been primarily a domestic poultry problem. Managed to get into wild birds somewhere in 2005, signaled the westward march of that virus, and that sort of seeded the problem for other continents, including Europe and Africa, but again had spared the Americas and Oceania. That changed for those of us in the Americas in late 2021, again through a change in the dominant virus circulating in wild birds. The virus became, again for reasons we don't understand, became that much more adapted for wild birds, much more of it in that environment, and subsequently we saw the spread then from out of this region through into Canada, and then subsequently down through the US, and then down into other parts of South America. Again, the Oceania currently is really the only continent that's unimpacted by this particular virus. Of course, the virus we're talking about now is the 2344B version of this virus. We can go into what that means later on, but that'll take my 5 minutes just explaining that. I just wanted to highlight the fact that this current batch of 2344B viruses, and I'm not just talking the virus in cows, I'm talking this group wider. So the ones that are, again, spread throughout Europe, spread throughout Africa, in certainly parts of Southeast Asia and in the Americas. It has properties that we haven't seen in these viruses before. To me, the biggest example of how this virus is different than anything else is just looking at the Americas. Since 1996, we had seen one introduction of this virus via birds into the Americas, into the continent. Came in at the end of 2014, was spread as the migratory birds made their migration south, but never made it past summer. So certainly never made it to that migration back up. So one introduction in all of those years. Since the end of 2001, we've seen at least 6 separate introductions of this virus into the continent now, both from both from west into east and also east into west. So again, I think that really, to me, highlights how the ecology, epidemiology of this virus in birds is so much different than anything we've seen. Of course, I don't think we need to spend much time on this. We're, I think, all too familiar with this particular version is impacting bird species. Here's some buzzards just not far out of Atlanta in Georgia, impacting bird species that haven't experienced this virus before. We're seeing the introduction, I'd say the spike in detections in mammal species. Again, we can argue whether that's a property of the virus being more infectious for that host, or just the fact that in many, many more of these infected birds around, so much more exposure risk between these wild mammals. We're also, of course, this virus is teaching us new things about where influenza viruses can replicate. If you look at any textbook of influenza, you certainly won't see dolphins, you won't see porpoises, and you probably won't see cows as host of that virus. But again, now we know that these are susceptible to influenza, and that's all due to the spread of the 2.3.4.4 Also, just want to point out here, one of the models we use for risk assessment for influenza viruses is the ferret model. The 2.3.4.4 in general are very, very virulent in this model, so cause a very severe disease, high neurologic involvement. And we also see some transmission in this model. So transmission between infected ferrets and either a direct contact animal and sometimes at airborne contact animals as well. That is atypical for H5s. So there is some biologic properties of the 2.3.4.4 outside of sort of the ecology and epidemiology that is different. And they are different than everything else we've been dealing with since 1996. So gaps and opportunities, from a virologic perspective, looking at what the virus has done so far as it spread, as it infected foxes, as it infected dolphins, as it's infected cows and humans. There's, I think from a glass half full perspective, we're not seeing too many changes in that virus associated with mammalian adaptation, which is a good thing. We know roughly what to be looking out for. We haven't seen them, but at the same time, we haven't seen strong genetic signatures associated with some of these other events either, including the infection of cows, including that what were the changes that led to this virus being far more, I guess, fit for wild birds, and that sort of explosion in the late 2021 that brought the virus to the Americas. So with that, of course, but from a good perspective and opportunity, if again, this is a silver lining for sure, but we are now getting much more data on this virus in these different hosts, so we are starting to learn a little bit from that perspective. And I'm sure we don't see strong genetic signatures. That doesn't mean those signatures aren't there. We just don't know exactly what to look for. The big question, of course, where is this virus going evolutionarily, and what will drive the acquisition leader changes? In my mind, we know some of the changes that are needed for this virus to switch from being a bird virus to being a human virus. And it's pretty clear that this virus doesn't make those changes easily. Otherwise, we certainly would have seen far more spread. So something has got to change before this virus starts to pick up those mutations. And what are those changes? Is it replication in a different host? Perhaps it's getting into pigs. Is it just random mutations across the genome, or is it reassortment? We all know we're now headed into seasonal influenza season. What happens if we get reassortment of the H5 with seasonal flu viruses? Will that drive some of the changes needed for the key move over to humans? And again, I think from a vaccine perspective, the other big question is we don't fully understand what phenotypic changes might accompany that host switch as the virus moves into humans. It clearly has to change to move into humans. We know some of those changes that are required overlap with antigenic sites on this virus. So, if this virus does start to transmit between humans, will that virus have the same antigenic properties of the virus we have now? And what about the phenotype in terms of disease? Clearly from a human infection perspective, this virus, with a few exceptions, has caused a lot of mild symptoms. But again, we don't know if that's going to be the case with a fully adapted virus. So Rebecca, I'm going to stop there, and happy to answer questions when it comes time. Thank you.
Great, thank you so much for that. Marcela, should we just go straight into your presentation?
Thank you. I don't have a PowerPoint, so nice to be here. Thank you, Dr. Webby, for that introduction, and it's a very good segue into what I wanted to share with you today. So as we prepare for what may be a human pandemic, I want to remind us all that we are, since 2020 or late 2020, in an animal pandemic that is basically all world round except for Oceania at this moment. And I want to highlight the fact that it is present at this moment, causing outbreaks in the Southern Hemisphere and the Southern Oceans near Antarctica in several different locations, including now the Indian Ocean islands. So this threat is far from going away, and it is recurring as each season in the Southern Hemisphere returns with the movements of animals. And I want to remind you that in the Southern Hemisphere, we're heading towards summer. So it's kind of the opposite of what you would expect. In the old days, we used to have these peaks of influenza in the winter, and now the seasonal appearances are gone. And that is one of the main features of this ongoing panzootic. The other thing I wanted to mention was that why this is relevant, that we have an ongoing panzootic for the last 3 years or more, is that we are giving this virus millions of opportunities to evolve and change and go into different hosts. And we're seeing that experiment occur naturally. And we have this amazing biodiversity and this huge catalog of viruses that, you know, can mix and change and adapt in these different hosts. And that is kind of what we're seeing with this virus. And in 2005, when the virus started spreading out of its origins, we identified this, and we were in the Wildlife Conservation Society, identified an H5 virus and a whooper swan that then became the first candidate vaccine given to vaccine virus given to WHO for, in that moment, the first scare of a human pandemic. So I want to highlight this fact that wildlife in itself and the circulation of the virus in that group of animals provides us with a unique opportunity to get our hands on viruses as they're changing. And that is extremely relevant at this point, where the virus is going in different directions nearly in every continent. So there's independent evolutions of these strains going on now in birds and in North America, and it's different from Europe and it's different from South America. And if I may, I'll hone into South America, where I live and work. And we had there a very unusual situation where we had an introduction for the first time. As Dr. Webby said, we hadn't seen this in the past. And maybe because of that, most likely because of that, we have massive impacts on all our wildlife. So we've lost in excess of 680,000 birds to this point, only in the last year and a half or two, and nearly 60,000 mammals. And this is wild mammals, pinnipeds, especially marine mammals, where we had never seen this virus before. And interestingly, we have seen already how this virus, upon entering South America, actually evolved into different clades, one that continues to circulate in birds with minimal changes, and then another one that actually made several attempts of jumps into mammals and finally ended up having the capacity to very smoothly move with mammals all around South America, from north to south, and then from south to north along the Atlantic. And this has meant the geographic spread of a virus that we thought was a bird virus and now is moving quite easily amongst mammals. And, you know, maybe it is secluded into the coastal areas, and that's why we're not seeing a lot of issues in terms of human contact and human risk. But we still have this amazing opportunity of and the need, actually the urgent need, to tap into those resources to understand the genomics of this virus. And my key point here, I think, is that in terms of what we can do in the future, I wanted to say one more thing, is one thing we need is to understand what these viruses are. And for that, we need genomic level surveillance, because these changes at this point are very subtle, and we want to be very early aware of what these changes are. And just to give you one example, and I'm sure the discussion today will be a lot about equity as well, not only in access to vaccines later on, but now in the process or the early warning, is that if we only consider the Americas, North America is responsible for about 80 or 90% of the virus sequences are out there, even though it only has about 10% of all the cases in the Americas. So there's a huge disparity in the capacity that the countries in other parts of the world have to do genotype surveillance and even accessing these animals. When you talk about Marion Island at the point of South Africa, or when you talk about Crozet Island in the Indian Ocean, French territories, I mean, getting samples from animals that are there into a reference lab where you can actually do sequencing is extremely challenging. So maybe from a human health perspective, that is really hard to understand because we have the patients at hand, but the work that we do with wildlife is extremely complex. And I cannot just go and anesthetize an immobile, just get a serum sample from an elephant seal now to see if it has antibodies. I have to anesthetize a wild animal that suffered a great mortality last year, and I have to put it at risk, make it risk losing its baby when it suffered a 70% reduction in its population due to an outbreak last year. So just to keep all of these things in mind, our reality is very challenging in the animal and wildlife world. And I'll end by saying that we can only continue to provide the information needed to help human health and be prepared for a human pandemic and provide prototype viruses for vaccines or candidate virus vaccines for vaccines is doing more intensive surveillance and better surveillance, quality surveillance, and that will not happen unless there's more collaboration. This One Health framework that we talk about has to become real. There has to be north to south exchange. There has to be more equity in that space as well. There have to be more resources coming our way. We do all these things on a budget string. And ending, I think I will encourage us all to keep in mind that interventions in the animal space are only possible in domestic animals. We cannot intervene to help wildlife, and we should be expecting, beyond recovering viruses for our protection, we should be expecting cascading ecosystem impacts from what this virus is causing worldwide. We're talking about millions of animals dying due to this, and the possibility of entire ecosystems being upside down because of these losses into the future. So what the consequences of this are going forward are still unknown, and that will be, of course, something else that we'll have to deal with down the pipeline. So that's my contribution. Thank you.
Marcela, thank you for this. We're off to a pretty sobering start. So Jen, over to you.
Thank you so much. It's really a privilege to be with you to talk about this sobering topic. I'm going to focus, because there's been such great information shared already, just kind of focus on the kind of human epidemiologic perspective and sort of what I'm worried about. Obviously, as we heard, this virus is not a new threat. It's one that we've been tracking for several decades now. And one of the reasons that this has been something of such concern is clearly the worries about the potential for this virus to mutate, to become more easily capable of infecting humans, and then more easily capable of spreading between humans. Were it to gain those abilities, the abilities to infect and spread easily between humans, we would be in another pandemic. So obviously tracking it from a future pandemic risk. But it doesn't even have to cause a pandemic for us not to worry about the human health impacts of this virus. And in particular, seeing that level of severity among the infections that have been identified to date is another important reason to worry about this virus. I mean, when you see a case fatality ratio of 50%, knowing that surveillance is probably imperfect, knowing that we're probably missing some level of cases, you don't even usually see a 50% case fatality rate. So that really sort of distinguishes this virus as a human threat to those who are directly infected. It's quite a startling startling number. And, at least in the kind of early part of tracking this virus, there wasn't a lot of evidence suggesting widespread mild or asymptomatic infection. When you look at the serologic studies that were conducted earlier, there wasn't evidence of, entire populations being infected that would really drive down what you would expect a case fatality ratio or infection fatality ratio to be. So just to say this is quite a concerning virus on its own, even absent in its ability to cause a pandemic. Obviously, events have changed in the last couple of years, and in particular in the United States, first finding infections in cows, which hadn't been noticed before, and then subsequently in dairy workers who had exposure to infected cows. This is the study from the first outbreak in the US. It was a patient who experienced conjunctivitis. Here's actually the picture of the patient from the study. So just to see, when we say conjunctivitis, this isn't exactly, the kind of pink eye that your kid brings home from school, quite a serious case of it, but not necessarily infection that put someone in the hospital. That is, of course, heartening, but I still remain worried about the potential of this virus to cause severe infections. And I'm going to talk a little bit why. These are the human cases of H5N1 that have been reported to date in the United States. As you can see, this is really popping up across the country and causing infections in multiple places and in a continued fashion, meaning I think there was some hope perhaps in the ag sector at the beginning that this might kind of wipe through animal herds quickly. I don't see evidence of that. In fact, this thing still seems to be popcorning up across the country. We know from a variety of data sources that detection of infection is likely hindered by a number of things, including surveillance on farms. As of late, the human infections that have been identified are largely tied to whether or not they are working on farms in states that are conducting bulk milk testing. So testing the bulk, in bulk, sort of like pooled testing of milk being produced from farms. And when there's positives, they're able to sort of work back and figure out from where the farm, the milk, the contaminated milk may have come. But to date, only a handful of states participate in that bulk milk testing. It is expected to expand. So I anticipate that we will find additional human cases as a result. And that is quite important because there is evidence that that infections are going undetected, either because people have mild symptoms that they don't notice, or possibly because they don't have symptoms. There may be asymptomatic infection, or more likely in my mind is that there are deep disincentives to reporting infections in some of these workplace settings. I think one of the main points, though, I want to make is that of these cases that we have found to date, they have been predominantly mild in the United States, and we don't yet see evidence of sustained human-human transmission. In my view, we should expect limited transmission. If you look through the literature, there's actually quite a number of instances in which there's some evidence of potential limited human-to-human transmission. So perhaps very close household contacts, perhaps healthcare workers who are exposed to a sick patient in their care. This isn't to kind of raise, panic about that, but just to say it should be expected that we may find here or there transmission, and I think in responding to these outbreaks, take efforts to make sure we prevent household or other close contact transmission. One of the main questions about the cases that we've seen to date is why they have been quote unquote mild. And there are a number of hypotheses for this. I will say, I hope we can get into a conversation of this. In my view, they remain hypotheses. One is that perhaps the mechanism of exposure that the farm workers are experiencing may be different than how people have traditionally gotten infected with this virus. There's also a hypothesis that perhaps prior exposure to influenza, particularly maybe H1N1 influenza, which has been circulating since the 2009 pandemic, may be conferring in some people some level of protection. These are interesting hypotheses for which there are some interesting data that may support them. But I would say at this point, I don't think we have conclusive evidence of this. I would also caution an over indexing of hope on this, because even if it is true that prior infection does confer some protection against currently circulating H5N1 viruses, we also have to remember that not all of us have that same level of protection. Newly susceptible people are born every day. It may not just be if you've ever gotten H1N1, it may be more important which was the first influenza exposure you've had. And so there's just lots of unknowns and lots of open-ended questions. And so I am personally not assuaged in terms of worry about the potential for H5N1 to cause severe infections in the people who are being exposed to it. And those worries were definitely not abated, and on the contrary, they're really kind of accelerated with this most recent case that has been reported in Canada of a teenager who's hospitalized with H5N1 exposure, infection, no known exposure to farms, possible exposure to wildlife, but not yet known. This child is in critical condition. And, in my view, this combined with some of the other data that Dr. Webby talked about in terms of animal studies, I think we should remain concerned that people who are exposed to this virus and become infected could develop severe illness. And we should really, not wait for severe illnesses to occur to take action to protect people from that possibility. Right now, my biggest concern is in protecting farm workers who we know are experiencing this virus. This is proving to be sort of a persistent occupational hazard. And there are a number of challenges to protecting farm workers in these settings. PPE has been recommended, PPE has been made available. We know that PPE usage is not ideal. That is one challenge. Another challenge is there are disincentives to participating in surveillance programs on farms and disincentives to identify as having been infected. And so in my view, I think when we talk about vaccines, I think we need to talk about it in the context of how incredibly operationally difficult it is to use other forms of protective measures in these settings to prevent farm workers from, if they do become infected, from developing severe illness. And I'll just sort of end with this, which is to say that it's about to get a lot harder. And there are concerns about co-infection, people having H5N1 and then also being infected with human seasonal influenza and the potential for reassortment or further mutation of the H5N1 virus. Those I think are, real and important concerns that we should talk about. But I'm also concerned that operationally responding to H5N1 in the context of a high burden of other influenza A infections just makes it a lot harder to target our resources to differentiate, who has seasonal flu and who has H5. Knowing that they may have different potentials for severity, I think makes it important to differentiate those. But some of the surveillance that is happening now is happening in the context of a low expectation of finding influenza virus because it hasn't yet been flu season. And so once the volume of influenza A infections go up, it just makes it harder to differentiate, and really I think speaks to the importance of making sure there are H5 specific tests available so that we can more quickly discern between who has seasonal influenza and who has H5, which, given the kind of open-ended questions about the severity of those infections, I think is really important to know. So with that, I'll end there.
Thank you all, and we're relatively close to on our very strict time schedule, so we'll take that as a win for the first session. There are a bunch of questions that are coming in the Q&A portal. There's also some from the other panelists that are coming in as well. I'm gonna pose a few. I just want to say there are a couple of questions that have been coming in around recording. My understanding is that this is being recorded and will be made available. And I think that there's already deep interest in what's being said. Let me start. I have a question for Richard around the question about what you make of the reports of changes in receptor affinity that's been reported for the virus and the enhanced ability to spread between ferret cages. I guess, we'd like to gauge how worried you are.
Yeah. So again, to a bit more detail around that, we know influenza viruses from birds bind to slightly different receptors than influenza viruses from humans. And so there's been one report suggesting that some of these viruses from cows, at least one virus from cows, had picked up an ability to bind to the human virus receptor. So I think in general, that is not a common property of these viruses, luckily. We and others have looked at some of these in a range of different assays and certainly don't see that. So the viruses, whether they be in birds, whether they be in cows or dolphins, whatever, still have maintained that avian characteristic of binding to the avian receptor. Yes, I mean, nothing in biology is ever black and white, right? So yes, we may see some exceptions to the rule, but it's certainly not the dominant form of virus. So in my mind, 2,6 binding is something this virus, so that's the human receptor, does not have. So why is it transmitting between ferrets? So again, we use this as a predictor for transmissibility between humans, and H5s have not done this really in the past. I don't think that's a receptor binding change myself. I think these viruses grow like weeds, whether it's in the udder of a cow, whether it's in the upper respiratory tract of a ferret or the brain of a fox. They grow really, 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 really well. So I think what we're seeing in those transmission models, and this is a lot of hand waving, of course, is there's just so much virus in that environment that you can push the virus to transmit without it really picking up those more human mammalian virus markers.
Thank you for that. I guess Jen, you mentioned the teenager in British Columbia, and I guess a question for anybody who knows is maybe we can say a little bit more about what's known about the different virus that's causing the severe infection. My understanding, at least from the stat reporting, which was as deep as I've gone on this, is that linked more avian, less cow. But again, I don't know if you've been following this or have any more details.
I can maybe make a couple of comments on that. So yeah, this is certainly not the virus that's in cows. So it's a different form of the virus. It has the same H5 component. I think what's interesting about this group of viruses, and this is the group of viruses that seems to have caused the human infection in Canada, also the poultry and the swine and other infections in the Northwest, is that it's a different form. So that N1 component, so the H5N1, it is actually a different N1. So it's from wild birds, a different wild bird, not the one that came over from Europe originally. What that means for this virus, we don't know. But again, as I think tried to get across before we've seen, when we have seen major changes in what this virus does, it's usually preceded by some reassortment event where it's picked up a different combination of genes. So we're not good enough to predict what that means in terms of where the virus is going to go, but at least in my mind, it does range that, this, could potentially be signaling something different in this virus, but I guess time will tell. But yeah, pretty clear that is not the virus from cows.
Jen, you want to add anything about the Canadian case, or?
I don't think there's much to add at this point.
Yeah.
I mean, I think there are a lot of interesting ideas being suggested. I will just say, as an epidemiologist, I really caution about extrapolating too much from small numbers, because what you see in one person may apply to the next person, but it also may not. And so I think in all of this, not just sort of trying to understand what this one case means, whether it's the virus, whether it's the host, whether it was the way that the person was exposed, how much virus they were exposed to, lots of unknowns here. We also just don't know going forward what it means. And even in sort of the farm worker population that we're seeing, I mean, we're talking about a few dozen cases that we know of that are in a largely kind of young working age population. It's just hard for me to say how that's going to play out, and particularly how it's going to play out in other age groups, in other types of patients. And, we saw this in COVID. And just to remind us, if you have a virus, COVID, that just on the initial case fatality ratio calculations looks far less severe than what the initial H5N1 case fatality calculations were. We know that the majority of people who contract COVID, even pre-vaccine, didn't die, but a small percentage of patients do die. And when you have a lot of people exposed, it just becomes, a real big societal challenge. And so seeing something in a very limited number of people doesn't predict how it's going to affect the very young, the very old, two groups that we know are historically most vulnerable to influenza infections. People who may have different level of prior exposure to influenza viruses, et cetera. So I just really worry about over-indexing on a handful of data points.
I appreciate that. Marcela, let me turn to you. There was a question that was posed in the chat around whether the kind of catastrophic effect we're seeing in marine mammals right now, if you think it's related to the virus itself, or a little bit to variation on Jen's point, was this just something we missed before? Is this an issue around actually being able to do detection well?
Yes, thank you. That is a great question. And no, we monitor marine mammal populations very closely, and I've been personally working on that for the last 30 years. I know the elephant seals that died last year had never before been exposed to influenza A viruses, because I've been monitoring precisely for that for all this time. And that is the same worldwide. So there was the massive die-offs of marine mammals in the past have been morbilliviruses, and then now it's influenza viruses. And the only previous mass mortality, quote unquote, that killed about 400 animals, 400, was an H7 virus. And then we had a little outbreak here and there of H10, and then an H3, and then the occasional finding of an H1 virus, never high pathogenicity viruses. And everything like completely, shrinks in comparison to what we're seeing now. At only one site where I was last year, we documented 97% pup mortality. All the breeding pups that were born last year, 17,000 of them died within a couple of weeks. And I want to bring to this group's attention again, as Dr. Nuzzo was saying, we are in a soup of virus. I mean, there's virus everywhere around us at this point. And this is why I was trying to say we are in the midst of an animal pandemic. So this virus is circulating left and right in mammals and in birds, as far as we can tell, some of them not showing any signs of disease. And I have actually personally witnessed that. I've seen seagulls eating, elephant seal carcasses, and they were just fine, the seagulls. And the CT values in the elephant seals were as low as you can imagine. They were loaded with virus, and those gulls were just spreading it. So I just want to remind you that no, this has never been seen before in marine mammals, and I want to bring it to everybody's attention because this virus is transmitting between the marine mammals. In some cases, it has many acquired mutations that allow it to do that. And we're actually seeing it come back into birds, reversing some of those mutations, and we're trying to track that now. So I think that is why I emphasize that our ongoing surveillance and our reinforced surveillance of what genotypes and what the genetic composition of this virus is doing in wildlife is so relevant, because it's just everywhere around us. I mean, if we only look at what's happening in the domestic animal sector, I think we're missing, the biggest part of what this event is right now. Thanks.
Thank you. We have run out of time for our session. There are lots of questions that are coming in the Q&A portal, and hopefully we'll be able to try to address some of those offline after the panel has ended. I greatly, we had the task of trying to get a lot of information into a very short period of time. Very grateful to all of you for helping to set the scene and also raise some of the very real challenges that we have in front of us. So with that, I will turn back to the organizers, to Jesse and to the O'Neill team, and tell us what's next. Thank you.
Right, I think we'll have a brief break while the next panel gets ready, and probably there will appear on the screen, the meeting.